So, just to recap, we were just looking at convergent versus divergent evolution. And so we saw that homologous structures are a good example of divergent evolution. They are showing you things that at one time may have had a common ancestor and had similarities, but over time, because of different environments and different environmental pressures, they develop or they have varying adaptations. So that is divergent evolution, or they're getting further and further apart. Convergent evolution, where things look starting to look more and more alike or have more similar traits. Examples of that can be analogous structures or vestigial structures. So remember, vestigial structures are things that are inherited, but they no longer have a use. So possibly the appendix in humans, hip bones in whales and snakes, and other various things, traits that we may see in other organisms that don't really make sense why they would still have them, except for maybe their ancestor had a use for it. If we look at the bodies of the shark, the penguin, and the dolphin, we'll see that they all are very streamlined and they can move through the water pretty easily. And so even though the shark is a fish, the penguin's a bird, and the dolphin is a mammal, they're not closely related. Their similar environments give them similar traits over time because of what is favorable. So all of these are examples of convergent evolution. We looked at comparative embryology, looking at how during times of development, how different organisms that are not closely related do share some characteristics during development, and comparative biochemistry, actually looking at the DNA and protein sequences in order to determine how closely related those things are. So all of the differences that are accumulated within the group are called divergence. And over time, a brand new species that forms, that process is called speciation. We can have different things that cause types of selection. So we have directional selection, stabilizing selection, and disruptive selection. In directional, the dotted line is what is the normal. And this, one of the extremes of the traits are selected for. So for example, in this example, we have very short tails for this particular lizard, and very long tails, and then a lot in between. And in this case, it seems that the ones with the very long tails are more likely to survive because they look more like snakes and scare predators away. And the ones with the shorter tails do not survive. So over time, the tails of these lizards will become longer because those are the ones that survive and pass their traits on. And this one, for stabilizing, it's the medium trait that's selected for. And here it's the medium tail that has the better chance of survival because long tails drag them on the ground and slow them down, and short tails don't give them the same balance. And then here we see that for these squirrels, it's the short tails that help to keep predators from catching them, and long tails help the ones in the trees have better balance. So this is called disruptive selection because over time it's the medium-sized tails that aren't good either on the ground or in the trees. And so this one's probably the one that would actually lead to two separate species or speciation. So this is disruptive selection. And looking at classification, we are looking at how things are organized, and the broadest or biggest is called domain, and the most specific are species. Remember, this is taxonomy, or the study of classifying things. And the scientific way of naming different things are called binomial nomenclature. And the broadest, the domains, there's three main domains. We have archaea, bacteria, and eukarya, which lead to our 6 kingdoms here, where we're going to look at